Now the microtubules. Microtubules are non-branching, rigid and hollow tubes of protein. They can quickly disassemble in one location and reassemble in another. Where their function is needed, they can quickly reassemble in that area. And they have an average diameter of about 25 nanometers. They are the thickest of them. The microfilaments have the 5 to 7 nanometer diameter. Microtubules are having 25 nanometer, and the intermediate filaments are having 10 nanometer diameter. So they are the thickest. Now some of the biochemical aspects. Each microtubule is made up of 13 protofilaments. See, this is a microtubule, and this is a cross section of a microtubule, the transverse section which we have studied in the previous presentation. Okay, and it is made up of 13 units. We can see we can count from here: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So it has 13 protofilaments, 13 units. Now each protofilament is made up of polymerization by the two heterodimers, the two tubulin heterodimers. See, it is the heterodimer, that is the two components of the dimer. The two monomers are separate. The two monomers have separate biochemical properties, the alpha tubulin and the beta tubulin. Okay, and the polymerization is done in a slightly spiral manner. The polymerization is accomplished at the microtubular organizing center, which is toward the centrosome of the cell, which forms the mitotic apparatus, the mitotic spindle. Polymerization of the tubulin heterodimers requires some molecules and ions, as like that of actin filaments, that are the calcium ions, magnesium ions, and the guanosine triphosphate for energy purposes. Now the microtubule formed has two ends, a non-growing end or minus end, which should be located somewhere here, and a growing end or plus end, which is this. And to prevent the depolymerization, there are some special proteins which are the membrane associated proteins. These proteins prevent their depolymerization. Specialized proteins, the membrane associated proteins called the motor proteins, help in the motor transport by using ATP. Okay, these are the specialized membrane associated proteins which are called motor proteins, which help in the motor transport within the cell by using the adenosine triphosphate. They are of two types, kinesins and dyneins. Now the kinesins move the cellular materials, the organelles, vesicles, substrates, etc. from the center to the periphery, that is from the nucleus to the cell membrane. This transport is called the anterograde transport. Dyneins move the substances, the vesicles, the cell organelles from the periphery to the center, which is from the cell membrane to the nucleus, and this transport is mainly called as a retrograde transport. Okay. Now the functions. It maintains the cell shape. See, this is the hypothetical cell, and here are the microtubules situated. See, they are maintaining the cell shape. They help in the formation of cilia and flagella. The cilia and flagella in the human body help in the propulsive movements of mucus and some other materials. Now here is the basal body, and here is the stalk of the cilia, and inside it there are present the microfilaments. As we have discussed, it was the microtubules there. They are the microtubules inside. Now, they also help in the transport with the membrane associated proteins, the microtubules. This is the kinesin and the dynein. Kinesin moves towards the plus end or towards the periphery, and the dyneins are moving the materials towards the minus end or to the nucleus. They also help in the formation of the mitotic spindle, the microtubules. See, they are forming the mitotic spindle, which helps in the cell division. Now, the following intermediate filament is present in muscle cells: keratin filaments, neurofilaments, Desmin filaments, vimentin filaments, and microfilaments. If you have answered C, So you are correct, because these are the Desmin filaments which are present in the muscle cells. The structural unit of the intermediate filament is G-actin, tubulin, protofibril, kinesin, none of these. If you have answered C, that means you are very correct, because it is the protofibril which is the unit of an intermediate filament. The unit of the microtubule is tubulin, and the unit of the microfilament is the G-actin. The following membrane associated protein helps in the transport of vesicles from the cell membrane towards the nucleus: spectrin, gelsolin, kinesin, actin, dynein. Now the correct answer is E. Those who have corrected it, very good, because dynein protein transport the cellular materials from the periphery to the center, and kinesin move from the center towards the periphery. So here we are at the end. These are the references. If you have any questions, you can freely ask. If you have liked my video, please subscribe my channel and click the bell icon for the updates. Thank you. Allah Hafiz.